Hello, my name is Daniel Bacher. I'm from Oregon State University, and my project was investigating expression of the Chlamydomonas bicarbonate transporter LCIA in plant chloroplasts. I completed this project in the Hansen lab under the mentorship of Dr. Vishal Chaudhry. Rubisco is the enzyme in photosynthetic organisms which catalyzes the important first step in the Calvin cycle as part of photosynthesis, where carbon dioxide is converted to organic carbon. However, at atmospheric carbon dioxide concentration, this reaction is inefficient. This is because Rubisco is catalytically slow and gets confused when trying to tell the difference between carbon dioxide and oxygen. Fortunately for us, but unfortunately for Rubisco, oxygen is present at higher concentrations in the atmosphere. So with no method to increase carbon dioxide concentration, with no method to increase carbon dioxide concentration in the cell, Rubisco reacts with oxygen a lot, leading to a loss of fixed carbon and energy for the plant. This reaction is called photorespiration. As you might expect, at higher concentrations of carbon dioxide, Rubisco seems to be more efficient, reacting more with carbon dioxide and wasting less energy on photorespiration. Thus, if we could increase carbon dioxide concentration around Rubisco, if we could increase carbon dioxide concentration locally around Rubisco, we believe we could increase Rubisco's efficiency in fixing carbon and decrease photorespiration. Because Rubisco is so inefficient, most land plants dedicate a large amount of their resources towards photosynthesis and still lose energy to photorespiration. So increasing Rubisco's efficiency could also lead to improved growth. One strategy that some photosynthetic organisms have employed is to increase carbon dioxide concentration around Rubisco with the use of active bicarbonate transport. Bicarbonate can only cross membranes actively, unlike carbon dioxide, which can enter and leave through diffusion. So it's much easier to increase bicarbonate in different compartments of the cell, where it then equilibrates to carbon dioxide. Chlamydomonas, a photosynthetic alga, has bicarbonate transporters like LCIA on its chloroplast envelope, which bring bicarbonate into their chloroplast stroma. No known land plants express bicarbonate transporters on their chloroplast envelope or anywhere in their cells, so they represent an untapped potential strategy to improve photosynthesis in plants. LCIA is a carbon dioxide channel expressed on the plasma membrane of Chlamydomonas, which actively transports carbon dioxide in the cell in low carbon dioxide conditions. As a result, transferring LCIA into plant cells is likely to aid chloroplast bicarbonate transporters in their ability to increase carbon dioxide concentration in the chloroplast by increasing carbon dioxide concentration in the cytoplasm. The theory is further bore out by simulations that have suggested that adding bicarbonate transporters to the chloroplast envelope in plants would increase photosynthesis by 9 to 16%. So this summer, I was tasked with working towards investigating if this strategy can be used by plants to improve photosynthesis by working towards expressing LCIA and LCI1 in tobacco, which is an effective model system to develop strategies to improve a wide variety of crops. The work that I did this summer will help towards confirming if the theoretical analysis of bicarbonate transporters improving photosynthesis will come true in real plants. But how do we take a protein from one organism and express it in an entirely different organism? It takes several objectives being reached. First, I created a library of plasmids, which are circular pieces of DNA, containing Goldbrick's parts, which are various DNA sequences necessary for transgenic expression in plants. Next, I made expression cassettes from those parts, combining every necessary element together into a form that can be expressed in a plant. Finally, I transiently express the genes in plant cells with fluorescent tags that will allow for the understanding of the localization and expression of LCIA and LCI1 in plant cells. In the future, LCIA and LCI1 and other genes from Chlamydomonas and other photosynthetic organisms can be stably expressed in plant cells, which will allow for the assaying and understanding of the protein's effect on the plant throughout its life cycle. In addition, by stably expressing these genes in plant cells, we can actually answer the question of whether or not the bicarbonate transporter strategy will work at improving photosynthesis in plants. The Goldbrick strategy for molecular cloning was developed in the Hansen lab by my mentor, Dr. Chaudhry. It involves cloning 3 necessary elements for gene expression: promoter, gene sequence, and tag terminator construct, individually into vectors containing antibiotic resistance, each with restriction enzyme sites on either end of the element. Using those restriction enzyme sites, the elements are then assembled into single expression cassettes, which contain further elements important for gene expression, including another antibiotic resistance gene used for antibiotic expression in stable expression. These cassettes can then be used for single gene transformations, as was the case for my project, or they can be combined into multi-gene cassettes and used for multi-gene transformations. Multi-gene transformations will be important when many different proteins are trying to be expressed and characterized all at once, which is ultimately how we will take a protein from Chlamydomonas and express it in tobacco. First, I use PCR to amplify the expression element and add restriction enzyme sites on either end. After digesting the amplicon with those enzymes, I ligated it into a library vector containing ampicillin resistance. That had also been digested with compatible enzymes. I then cloned the resulting plasmid into E. coli. On the screen, you see an example of this process, the CAMV35S promoter, which is a popular promoter for transgenic expression in plants from the cauliflower mosaic virus. But this is the same strategy I used to create each GB2 part that I created this summer. The E. coli then acts as something of a molecular factory, taking the plasmid that we built and creating a bunch of copies for us to use. Once I had colonies on the plate, I validated them using colony PCR, which essentially just involves taking the bacteria off the plate and putting it into the PCR machine. Each dot you see on the plate represents a colony of genetically identical bacteria, which may or may not contain our plasmid, so we have to validate them. Sequencing primers on the backbone were used to confirm that the insert was present. The primers are close together in the backbone without the insert present, but once the insert is inserted, they amplify a section of DNA that is larger because they amplify the insert. So as you can see, the case of PCAMVN, the insert that they amplify is about 1 kb. So that shows up on our gel, and these colonies as well as these are confirmed, while this colony is not valid. So using this strategy, I created Goldbricks parts for the PCAMVN promoter, a full cassette for expressing kanamycin resistance, which is our stable expression antibiotic selection marker, the TNOS terminator, the yellow fluorescent protein tag, which will be used to visualize the cellular localization of LCIA and other proteins in transient expression, NUC2, which is a backbone containing tDNA insert borders, and the HA and Strep2 tags, which are small protein tags that will not affect the function of the protein, but can be used for characterizing the protein in stable expression. These elements, when combined with the codon-optimized gene sequences that we have, can be used to characterize the localization and function of the proteins in tobacco and Benthamiana. Each of the proteins is predicted to have the potential to increase photosynthetic efficiency in plants by improving carbon dioxide concentration around rubisco. LCIA, as I've discussed, is a bicarbonate transporter on the chloroplast envelope, as is CIA8. LCIA, again, as I've discussed, is a carbon dioxide channel, where HLA3 is also on the plasma membrane like LCI1, but it is a bicarbonate transporter. And then Flavaria bidentis BCA3 is a carbonic anhydrase, which converts CO2 to bicarbonate, and it is present in the cytoplasm of Flavaria. Overexpression of Flavaria BCA3 in Arabidopsis thaliana has been shown to increase photosynthetic parameters and have a positive effect on growth. Eventually, each of these genes will be expressed in tobacco using the same method that I have outlined. Each of them will provide an opportunity to investigate how a protein from another organism can improve photosynthesis in plants. I decided to begin by transiently expressing LCIA and LCI1 in Benthamiana to visualize their localization. We predict that they will localize the same in plant cells as they do in Chlamydomonas, But it will be important to make sure before we move forward with stable expression. So to this end, I created expression cassettes with the PCAM-VN promoter, the LCIA and LCI1 coding DNA sequences, and the YFP tag. But how did I do this? First, I linearized the gold bricks part plasmid for PCAM-VN using these two restriction enzymes, and ligated the coding DNA sequence for LCIA downstream of the PCAM-VN promoter and the YFP tag downstream of that, which you can see the full expression cassette here, the PCAM-VN promoter, the gene sequence, the YFP tag, and the terminator. And I put them into E. coli and again validated them with colony PCR. But I still needed a way to express the genes in plants. Just injecting DNA or plasmids from E. coli into the plant will not allow the gene to incorporate into the plant genome or allow the protein to express in the plant cell. So I needed to do agrobacterium-mediated tDNA transfer. First, I needed to move the expression cassette into a form where it could be incorporated into the plant genome. I created a vector, which I called nuke2, which contained tDNA borders for agrobacterium, here and here. Anything inside of those borders will be injected into the plant nucleus and expressed in the cell when the plant is transfected with agrobacterium. Inside the borders were restriction sites that were compatible with the sites at either end of my expression cassette, here and here. So when we use those restriction sites and use them on our expression cassette, that expression cassette will go into the plasmid. So I cut nuke2 with those expression sites and cut my expression cassette with those expression sites and put it into the nuke2 promoter. So here we have the tDNA borders, and you can see that inside of the tDNA borders, we have that expression cassette. Our promoter is here, gene sequence, YFP, and terminator. Once I had the plasmid, I transferred it into agrobacterium by electroporation, and then into the plant leaf by syringe infiltration. To do this, I created a suspension of agrobacterium cells transformed with the expression cassette plasmid and literally injected it into the leaves. This is not me, but you can see an example of this here. This allows fluorescent tagged LCIA to temporarily express in the plants a few days later, which can be visualized with fluorescence confocal microscopy, much like the image you see on the bottom right. In summary, I created a library of parts necessary for gene expression in tobacco and expression cassette with the elements necessary for expressing LCIA and LCI1 tagged with YFP and benthomiana, which are immortalized in the Hansen lab in glycerol stocks. I also did syringe infiltration with LCIA tagged with YFP and benthomiana leaves. And we hope that fluorescence images will be available soon to confirm if LCIA really is expressed in the chloroplast envelope in tobacco. In the future, this methodology can be repeated to visualize YFP-tagged LCIA, LCI1, and other potential photosynthesis helper proteins, including the ones that I've already listed, in tobacco. And the GB2 parts that I have created can be utilized to stably express all these genes in tobacco to help further the understanding of how these genes may improve photosynthesis in tobacco throughout its life cycle. I would like to acknowledge the sources of funding for this project and my internship, the NSF and USDA, as well as the Hansen Lab plant researchers, particularly Dr. Chaudhry and the principal investigator, Dr. Maureen Hansen. I'd also like to thank Cornell University and the Department of Molecular Biology and Genetics and the Boyce Thompson Institute for hosting me. Any questions about this presentation should be directed towards me, and I can be reached at the email on the screen. Thank you.